Hello there. Welcome to Fundamentals of Biology Lesson 9, where we're going to be talking about cell membranes. So this follows on nicely from the last session where we looked at cell structures, where we had a look at prokaryotic cells and eukaryotic cells. And then we had a focus on plant and animal cells, so two types of eukaryotic cells. One of the structures that we discussed in that session that we find in all cells, whether prokaryotic or eukaryotic, is the plasma membrane, sometimes referred to as the cell membrane. And these play an incredibly important role in how individual cells do the things that they do, but also how cells come together to form tissues, which enables us to form organs and then organ systems. So cell membranes are a really important component of all cells on the planet, so they are worthy of individual investigation. So that's what we're going to be doing in this session. So by the end of this session then, you should be able to identify the key components of cell membranes. So we've talked about phospholipids in the past, and as we'll see, they are a main structural component of the plasma membrane, but there are also many other components in there. So you'll need to be able to identify those key components, and then you should be able to discuss the role of, or the roles of each of those membrane components. And then also, as we'll look at as we go through, there are several different ways in which things can get across the cell membrane, so how they can get into the cell or out of the cell. So we'll go through each of those four modes of transport across the cell membrane, so you should be able to talk about them and list the similarities and differences between them. So that's the plan of attack for this session. So let's start off by talking about the general functions of the cell membrane or the plasma membrane. I should probably point out at this stage that the cell membrane and plasma membrane, they are exactly the same thing. People tend to use those two terms interchangeably. So just look out for when I do that. I quite often flip between the two. So one minute it'll be the plasma membrane, and one moment it'll be the cell membrane. So just be aware that they are the same thing. The cell membrane is very, very different to the cell wall that we looked at in the last session. So remember, the cell wall is a rigid structure that we find around plant cells that's made of cellulose, but we also find it around fungal cells where it's made of chitin, and we also find them within prokaryotic cells, so bacteria and archaea, where it's made of peptidoglycan. So the cell wall is a rigid structure. The cell membrane is this sort of semi-fluid, largely made up of phospholipid membrane that we're going to be discussing today. So what are the general functions of the cell membrane then? Well, first and foremost, they separate the inside organelles from the external environment. So why is that important, you might ask? Well, once you've got a plasma membrane in place, it means that you can have chemical reactions and various organelles that can go about their business unaffected or uninterrupted by what's going on outside of the cell. That can help protect them from physical damage, but it can also prevent them being interrupted by different chemical processes. So this separation, so you've got a separate inside or internal and external environment, is one of the main functions of the cell membrane. They also act as gatekeepers, so they can control and regulate what gets in and out of the cell. And as we'll look at as we go through, proteins within the membrane play an incredibly important role in this regulation or control of substances getting in and out of the cell. So that will be an important component of cell membranes that we look at. They also are an important place for certain recognition and information tags to be found. So we'll find these in the forms of glycolipids and glycoproteins. These are little information tags that are attached to the outside of the plasma membrane, and they enable the transfer of information between cells so they can recognize each other. So cells can recognize things like pathogens, but also can recognize substances that they want to allow in. They also provide structural support. So in the last session, we talked about the cytoskeleton, so the microfilaments and microtubules. Well, as we see as we'll go through here, we can see that particularly microfilaments are often attached to proteins on the inside of the plasma membrane, and in combination, they can provide almost like a scaffolding structure that helps to provide support for the cell. I mentioned just now that cells will come together to form tissues, and then tissues will form organs, and then organs work together within organ systems to enable complex life to exist. And those cells are attached to each other via the plasma membrane, or particularly molecules that are attached to the outside of the plasma membrane. So we'll look at those in a bit as well. So what we can see on this diagram then is a close-up of a section of plasma membrane. So you can see that there's quite a lot going on there. It's quite a complex structure. As you're probably starting to realize now within biology, the closer that we start to focus in on something, the more complex and the more complicated it gets. It's one of the really interesting things about biology and cytology, so the study of cells, is that the more our techniques and our technology for studying cells gets better, the more incredible things we're finding about it, and the more complex and complicated it all appears. So here we can see a nice diagram of a plasma membrane, or a section of it. And when we start to examine it in more detail, we can see that it's made up of three main types of molecules. So we can see that lipids, proteins, and carbohydrates all work together to make up this structure. And here we can see the relative percentages of each of those within a typical cell. So we can see that there's about 42% lipids, and as we'll see as we go through, it's phospholipids, but there are also other types. And then there's also proteins at about 55%, and then there's a small amount of carbohydrates, so about 3% of those. But let's just highlight a couple of the main features that we'll examine over the coming slides. So way back in the session where we talked about lipids, I finished that section off by talking to you about phospholipids. So if you remember, those are molecules that are very similar to the triglycerides. So they have a glycerol molecule with two fatty acid chains attached, but instead of a third fatty acid chain, they have a phosphate group, which makes them ambivalent towards water. So they have hydrophilic heads and hydrophobic tails. And I said that they make these structures called bilayers. And that's what we can see here. So if we look at all of these grey-blue circles here, this is the phospholipid bilayer. The little circles here are the hydrophilic heads. So we can see that we have a whole layer of hydrophilic heads pointing out of the cell. That's what's meant by this extracellular fluid, this fluid that surrounds the cell. So we can see the layer of hydrophilic heads, and then we also have a layer of hydrophilic heads pointing into the cell, so into the cytoplasm. In between those two layers of hydrophilic heads, we have two layers of hydrophobic tails. So you remember the hydrophobic tails of a phospholipid are made up by the fatty acid chains. We also get these proteins, so these big purple blocks here. These are proteins. These proteins that span the entire width of the membrane. These are called integral or transmembrane proteins. And I've got that on a slide in a sec. What we also find, if you look where I'm pointing with the cursor, you get these smaller proteins that are just on the inside of the phospholipid bilayer. So they're attached on one side. And these are called peripheral proteins. And these play a really important role, as you can see here, for attachment sites for the cytoskeleton. So you can see microfilaments attaching there, and that helps to offer and provide structural support. So there are a couple of different types of protein. These little yellow bits that you get in between the phospholipids, well, they're another type of lipid. They're one of the steroid lipids that we talked about in a previous session called cholesterol. And then pointing out of the membrane, then attached to the outside of the plasma membrane, we have these glycoproteins. So that's a protein covalently bonded to a carbohydrate. So glycoprotein, and then here we've got a carbohydrate bonded to one of the lipids in the membrane. So these make up glycolipids. And those are important for the transfer of information and the attachment of cells to form tissues. So I'm going to go through and talk about each one of those in a little bit more detail on the coming slides. But I just wanted you to look at this diagram to get an overall feel for the plasma membrane of a cell looks like. One other point that I wanted to make as we're looking at this diagram is that the plasma membrane of a cell is quite a dynamic environment. It's sometimes referred to as the fluid mosaic model. And it's called the mosaic model because when you look at it, all of these proteins sort of embedded within it look a bit like a mosaic. So that's why it's referred to as the mosaic model. And the reason it's referred to as the fluid mosaic model is because it is fluid. These proteins are not fixed in position. They are constantly moving around within the phospholipid bilayer. So it's a very fluid structure with these proteins. You could kind of think of them as like moving around like a ship on the sea. They're kind of just moving around constantly. So it's sometimes referred to as the fluid mosaic model. When looking at the main structural components of the plasma membrane, then it makes sense to start with the lipids. So let's have a quick run through of the different types of lipids that I just mentioned on the previous slide, and we'll look at them in a little bit more detail. So when we break the lipids down that make up the plasma membrane, we find three main types. We find the phospholipids that we've already mentioned. So phospholipids, they're the major or a major component of cell membranes. And you get these two layers that make up the phospholipid bilayer. So you have these hydrophilic heads. So remember, hydrophilic is water loving. So these parts of the phospholipid bilayer will readily interact with water. And then in between those two layers, meeting in the center of the plasma membrane, you've got the hydrophobic or fatty acid chains. So this is a very hydrophobic area. So water is not going to be very comfortable in there at all. So this phospholipid bilayer makes up the main sort of structural component of the plasma membrane. So those are the phospholipids then, the hydrophilic heads on the outside and the inside. They will associate with water. And then the hydrophobic tails in between those two layers of hydrophilic heads. Glycolipids, as I mentioned previously, are lipids covalently bonded to carbohydrates, and they act as a recognition site for specific chemicals, as well as attaching cells to one another to form tissues. So we get this transfer of information and also as attachment sites between cells. So they may make up just a small part in terms of percentage of components of the plasma membrane, but they do play an incredibly important role. The one that I mentioned as well is the steroid lipid that we find within the plasma membrane, and that's cholesterol. Cholesterol acts as a kind of temperature buffer for the membrane. So if we look at the little diagram here, you can see these phospholipids making up the bilayer. So we've got these two layers of phospholipids, and then you can see the cholesterol sitting in between the phospholipids. And they play an incredibly important role in maintaining the fluidity of the membrane. So I mentioned previously that the plasma membrane is a fluid structure. It's constantly moving, and those proteins are moving around within it. And that fluidity is really important in enabling or controlling what can get in and out of the cell. If the membrane becomes too firm or solidifies, then the cell is not going to be able to control what gets in and out. And likewise, if it becomes too liquid, if it sort of falls apart, then obviously the cell will lose its structure. So what cholesterol does is it sits between the phospholipids and almost acts like a buffer. So if the phospholipids start to solidify, if they want to try and get close to each other, they cannot get close to each other because the cholesterol is sitting in between them. So it prevents the plasma membrane from hardening, if you like. And likewise, if the membrane were to start liquefying, so if it's exposed to warmer temperatures or something like that, so you've all probably put some butter in a frying pan. So you take butter out of a fridge where it's really cold and it's solid. You take some butter and put it into a frying pan and heat it up. It liquefies. So that process, in theory, could happen with the phospholipids within our membrane. What cholesterol does is it sits between the phospholipids, preventing them from getting too close to each other, so prevents them from solidifying, but it also holds on to them and prevents them from separating. So this is what we mean when we say that they act as a kind of temperature buffer. They help to maintain the optimal fluidity of the membrane. So we're often told that cholesterol is bad for us and we shouldn't have it. Well, cholesterol is essential for our lives. So cholesterol is important for this process, obviously, for maintaining the fluidity of our cell membranes, but it's also an important precursor for things like vitamin D, but also estrogen and testosterone. So cholesterol gets a bit of a bad rap. On to the next of our three groups of molecules that we find, and then we've got the proteins. So proteins are, in terms of percentage, so we saw earlier that they make up 55% of all of the structures within the membrane, so they make up the largest component. As I've mentioned, we often refer to the plasma membrane as a fluid mosaic model because the membrane is this mosaic of different proteins embedded within the phospholipid bilayer, as you can see here. So these are all different types of proteins, all of these big purple blocks here. Proteins are one of the main structural components that will determine the function that cell will actually have. Because as we're going to explore as we go through this session, it is the proteins that have a big role to play in determining what can get in and out of that cell. So as the proteins will determine what can get in and what can get out, they will ultimately determine what functions that cell will be able to carry out. And I mentioned this on the original slide looking at the diagram, that when we get these proteins here that span the entire width of the membrane, so part of them point into the cell and the other end points out of the cell, these are referred to as transmembrane proteins. And I'll give you some specific examples of these, or I'll show you where they're important over the coming slides. And then these ones that we find just on the inside and some we find just on the outside, these are referred to as peripheral proteins. So they're not embedded within the membrane, they're just on one side or the other. And these are really useful for attaching appendages to. So whether or not you're going to have like a glycoprotein, so you can covalently bond a carbohydrate, or whether you're on the inside and you attach your microfilament fibers of the cytoskeleton to provide structural support. So those are the transmembrane and peripheral proteins. I probably won't mention the peripheral proteins again, so just remember that they are there largely for structural support and for attaching appendages to. The transmembrane proteins will become really important as we go through the session though. Just to finish off this section, we can have a look at carbohydrates. And carbohydrates only make up a really small amount of molecules that we find within the membrane, but they do have some important functions. So the way cells recognize other cells is by binding to surface molecules. And those molecules will often be carbohydrates. And we can see some here attached to these proteins. Typically, they can be transmembrane or peripheral proteins. So we'll have these carbohydrates attached to make those glycoproteins. And this not only enables the cell to detect certain molecules and certain compounds, but it also enables cells to recognize other cells around them. So a cell's ability to distinguish one type of neighboring cell from another is important. That helps cells to sort themselves into tissues, but it also enables cells to reject foreign cells. So we could be looking at things like viruses or bacteria, so pathogens. It will enable the cell to identify those and reject them so they don't let them into the cell. So although carbohydrates only make up a small percentage of the material in the plasma membrane, they really do play an important role. And I've mentioned this previously, that the membrane carbohydrates are usually covalently bonded to lipids or proteins, and they will form molecules that we call glycolipids or glycoproteins, depending on what's bonded to what. Now that we've had a look at the main structural components of the plasma membrane, we can start looking at how it all comes together to control what gets in and out of the cell. So we've talked about the fact that cell membranes control the entrance and exit of substances. And what we find is that some substances can pass through the plasma membrane easily. And we'll talk about some examples on the next slide. But other molecules will need assistance. And some things can't pass through at all. So this ability for the cell to control and determine what gets in and what gets out is called selective permeability. So if you've ever studied biology before, you might often refer to this as partial permeability. But I never really liked that term very much, the partially permeable membrane, because it's not really accurate. A partial permeability just would mean that it's a very, very passive thing where some things can either get in or they can't. Whereas a plasma membrane is much more selective than that. It's actually choosing what can get in and get out based on the structure of specifically the transmembrane proteins. So it's much more accurately termed selective permeability. So that's what we're going to have a look at. I've got a little animation here, so hopefully this will work. So what we've just seen there are some examples of things that often go in and out of cells. If we notice this lipid here, a lipid was able to pass freely through the phospholipid bilayer. And that's because lipids are non-polar. They don't carry that electrical charge. So they can get through these hydrophobic tails here. So they can pass through completely freely. Sort of larger, more complex molecules like sugars, they're unable to. They cannot get through the phospholipid bilayer. The same with salts and the same with ammonia. But what we notice with these, particularly ammonia up here, it passed through these cylinders here. And you know what these cylinders are because they are the transmembrane proteins that we talked about just now. So notice how they acted like a sort of channel that enabled these things in and out of the cell. Instead of passing directly through the phospholipid bilayer, which they cannot do, instead they can actually travel through these transmembrane proteins. Over here we've got water. Now water's a bit of an oddity. So a lot of very, very small molecules can actually pass directly through the phospholipid bilayer. So things like carbon dioxide and oxygen, they can just pass freely through because they're so small. And water's a really small molecule as well. And its problem though is this hydrophobic center, these hydrophobic tails in the middle of the phospholipid bilayer. Water really struggles to get through there. So you've got the fact that it's a really small molecule making it advantageous for passing through there, but it's still, so water molecules can get through the phospholipid bilayer, but because it really struggles because it's all hydrophobic in here. So as we'll talk about in a bit, water also relies on the special transmembrane proteins to enable it to get in and out of the cell in the quantities that it's required. The structure of the plasma membrane then is what controls the entry and exit of molecules. Specifically, we're talking about those embedded transmembrane proteins, but also the fact that we have those hydrophobic tails in the middle of our phospholipid bilayer. And as I previously mentioned, really small molecules such as carbon dioxide and oxygen, they can just diffuse very, very freely in and out of the cell through the phospholipid bilayer. But if you have really large molecules, so certain carbohydrates and things like that, or if they are polar, so we saw again, so carbohydrates, but also things like salts and ammonia, these molecules and compounds that have an electrical charge to them, they aren't going to be able to pass through that hydrophobic portion of the plasma membrane. So how do molecules move between the intracellular, so that is the inside of the cell, and the extracellular, that is the outside of the cell, fluid or environment? So how do molecules move between the inside and the outside of the cell? That's what we're going to explore next. So movement can occur directly across the membrane, as we just talked about, like oxygen and CO2, or they can go through those protein channels. But either way, things will move in and out of our cells via one of four methods or modes of transport. And they are diffusion, facilitated diffusion, osmosis, and active transport. As we go through this next section of the lesson, I want you to keep in mind, and you can actually, if you've written these down, you can just circle number 1, 2, and 3. So diffusion, facilitated diffusion, and osmosis. Because these three share something in common. And that is, they all belong to something called passive transport. So they are all three types of passive transport. Now, passive transport means that the cell does not have to expend any energy for these three to happen. Diffusion, or sometimes referred to as simple diffusion, and then facilitated diffusion and also osmosis, all happen naturally. They are natural processes, a bit like gravity. If you have a ball at the top of a hill, you can just let that ball go and it will automatically and naturally roll down the hill because of the effect of gravity. You don't have to expend any energy to roll that ball down the hill. It's just going to happen. Diffusion, facilitated diffusion, and osmosis work in a similar process or in a similar way. They happen for free. Number 4, active transport, is the opposite. So active transport moves things, as we'll look at in a bit. It's more like moving that ball from the bottom of the hill to the top of the hill. Now you're going against gravity, so you have to invest energy to do that. And there are multiple ways you could get the ball up the top of the hill. You could carry it yourself, you could throw it, you could put it in a taxi and send the taxi up to the top of the hill. Lots of different ways, but every single way you do it, it will require that you spend energy to do it. And that's active transport. And when cells carry out active transport, they have to spend energy to do it. And they spend energy in the form of a molecule called ATP. And we mentioned ATP in the last session when we looked at mitochondria. So they are the powerhouses of the cell that generate ATP. So we have diffusion, facilitated diffusion, and osmosis that are forms of passive transport. They happen for free. And then we have active transport, which requires the cell to invest energy or spend energy to carry out. We need to go through each one of these 4 and talk about them in a little bit more detail. And they are important because, well, they help you to understand many life processes, but they will also underpin a lot of the other topics that we cover in this module later on, the terminology. So in later lessons, I'll be using the terms diffusion and active transport quite a lot, and also osmosis. And you'll also find that in some of your later studies as well. So if you are studying anatomy and physiology, or you are studying specific groups of animals, it's very difficult to talk about certain life processes, such as how the respiratory system, so how things like lungs and gills work, how kidneys work. So all of those really important topics that we cover in anatomy and physiology, it's very difficult to talk about how they work without having an understanding of diffusion and active transport and also osmosis. So it is really important that you follow this through and go back over it if you need to, because these are really important topics. Let's start by looking at diffusion, sometimes referred to as simple diffusion. And this is defined as the movement of molecules or ions, so it's just the movement of molecules from a region of high concentration to a region of low concentration. And we will often say that that is movement down a concentration gradient. So to simplify that, things will move from an area where there are a lot of them to an area where there are not very much of them until they are evenly distributed. Okay, so that's what we saw on this little animation. So I'll go back and then I'll restart the animation. So you can see how these little balls are moving from left to right until they are evenly distributed. And that's simple diffusion. So an example of this, where it's really important for life, is simply the movement or diffusion of oxygen from our lungs. So when we take a nice big mouthful of air, that air is high in oxygen and low in carbon dioxide. It goes into our lungs, and then oxygen will diffuse down a concentration gradient from our lungs into our blood. So the blood that is brought back to our lungs is low in oxygen because our body has used all the oxygen up, but it's also high in CO2 because the blood has gathered CO2 as it's been moving around the body. So the blood moving to our lungs is low in oxygen but high in CO2. The air that we breathe in is high in oxygen but low in CO2. So we have concentration gradients there. And oxygen will diffuse down its concentration gradient, so from our lungs into our blood, and carbon dioxide will diffuse down its concentration gradient from our blood into our lungs. So that is a nice example of simple diffusion happening. And remember, this is a passive process. It happens naturally. Our bodies and our cells do not have to expend any energy for this to happen. Let's have a little look at an animation for simple diffusion then.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So that was simple diffusion then. The next one we can look at is facilitated diffusion. So facilitated diffusion is still a passive process. It does not require any energy expenditure by the cell. It happens for free. But it happens with molecules that are unable to pass freely through the phospholipid bilayer. So if you cannot pass through the phospholipid bilayer, you need help. You need to ideally the help of one of those transmembrane proteins. So when we have large molecules, so we can talk about things like glucose, but also certain amino acids and even some smaller ions that cannot pass across the phospholipid bilayer, they can get help, they can get facilitated, if you like, through these channel proteins or through these transmembrane proteins. So an example would be something like this, which is a protein channel. So we can see here they can occur through hydrophilic. So these are water-filled channels. So I've put there as an example water. So we get a type of channel protein that are called aquaporins, and they are just proteins with a hole through the middle, essentially. That is made of, you remember, proteins are just made up of amino acids. So we would have amino acids within the middle of the protein that are hydrophilic, so water-loving, and that means now that water can pass freely through these channels, enabling it to diffuse freely in and out of the cell. They wouldn't be able to diffuse in anywhere near the same rate directly through the phospholipid bilayer. So this is a type of passive transport, but for it to happen, they need the help of these channel proteins. So that is why we refer to it as facilitated diffusion. The proteins are facilitating, they're helping the diffusion of water in and out of the cell. We also get another type of facilitated diffusion where instead of using protein channels or channel proteins, we're actually using carrier proteins. So you can see an example over here. And a good example of this is how glucose gets into things like red blood cells. So that will rely on facilitated diffusion using specialized carrier proteins. And these proteins will actually change shape to almost throw the glucose into the cell. It's not really throwing, but the glucose can come in, they can attach to the protein, which then causes it to change shape. The forming of the bonds between the glucose and the protein will cause the protein to change shape, which will then move it inside the cell. So the cell, again, is not expending any energy from this to do this. So it's a form of passive transport. The difference here is that you've got this channel protein that just allows water to flow through. And then you've got these carrier proteins that are sort of physically changing shape to help glucose diffuse into the cell. So that's facilitated diffusion. And remember, it's a passive process. The third type of passive transport then is osmosis. This is a particular special case that we need to take a little bit of time with. So osmosis, though, remember, is another form of passive transport. So this will happen freely. The cell doesn't have to expend energy to do it. But it will underpin a lot of really important processes that we'll go on to look at later in the module. There's a whole topic that we'll look at called osmoregulation. This ability for animals and other organisms to regulate their water and solute balance. So just to simply put it then, osmosis is the diffusion of water molecules. So it is specifically, when we're talking about osmosis, we're specifically talking about water. So it is the diffusion of water molecules from an area of high concentration to an area of low concentration. So so far it's just like diffusion, so the movement from high to low, but we're just dealing with water. But then comes this other bit, so it's the diffusion across a selectively permeable membrane. So what we're talking about here is specifically the diffusion of water across a selectively permeable membrane, such as the cell membrane. The simple way of looking at this is, if you have a lot of water molecules on one side of the membrane and not very much on the other, water will diffuse down that concentration gradient until it is evenly distributed on both sides of the membrane. Now that's all well and good, but what we find when we actually look at nature is that we very rarely find examples of pure water. There are usually an awful lot of things dissolved in it. So water is an incredibly versatile solvent. You can dissolve a lot of different things, from sugars and salts and amino acids and all kinds of other things in it. And those things that dissolve into water are what we refer to as solutes. So solutes, like I said, can be anything. So anything that will dissolve in water. So typically I'll be referring to salts and sugars and amino acids as we go through though. Solutes, so those things that dissolve in water, have a big impact on how water diffuses across our membranes. So there is a second way that we can define osmosis. The first way is what we've just covered. So it is the diffusion of water molecules from an area of high concentration to an area of low concentration across a selectively permeable membrane. However, we can also describe or define it as the diffusion of water from an area of low solute concentration to an area of high solute concentration. So why is that important? Well, let's have a look at this diagram at the bottom of the slide here. What we can see is just a little experiment that's made up. So this section through the middle here is going to act as our selectively permeable membrane. So you can think of this as the cell membrane. And then the little blue circles here are our water molecules. And then our big green circle or ovals here are going to be sugar, so sucrose molecules. Now, if we were to remove all of that sugar from there, so we've just got water in here, what you would find is water would diffuse down its concentration gradient until it is evenly distributed on both sides of the membrane. So there would be no difference between it. But now what happens when we add our sugar, that a lot of those water molecules will be attracted to that sugar as they dissolve it. So those water molecules, a lot of them will be basically stuck to each of these sugar molecules. What that does is it locks up a lot of the water molecules, so they are no longer free-floating water molecules that are able to diffuse. Now, we would say that this side of this experiment here has a high solute concentration. A lot of the water is locked up. It's attached to these solutes, these sugar molecules. So basically, there are a lot more free water molecules on this side than there are on this side, because a lot of the water is locked up. It's no longer able to diffuse or pass through this membrane because these sugar molecules now are just too big. So in which direction will water now diffuse? Well, it's going to diffuse, if we go back up to this definition, water will diffuse from an area of low solute concentration. So if we look at the left-hand side here, there are no solutes in here, so a low solute concentration. So water will diffuse from an area of low solute concentration to an area of high solute concentration. So the water will diffuse from left to right in this diagram. So it is going to move towards the higher solute concentration, because water is just trying to balance out the distribution of its water molecules. Because so many of the water molecules on the right-hand side here are locked up with that sugar, with that sucrose, they are no longer free, so they're no longer classed as part of those free water molecules. So that plays a really important role in many of life's functions, and it's going to become an important topic that we discuss in later sessions. We're constantly taking in solutes, so salts and sugars and amino acids in our diet, in our food. So we are always gaining those solutes. And then also we're having to use water in our urine to flush out waste products. And we're also losing water through evaporation and through sweat. So we have to drink water to help balance out our water and solute. And this is a process called osmoregulation, this balancing of water's salt content in our blood, in our plasma, in all of our tissues, and inside of our cells. So if you can imagine, if you've got your red blood cells, for instance, if your blood plasma, so the fluid, the liquid in which those cells are moving, has a higher solute concentration, so is saltier than the inside of the cell, well, what's going to happen to them? Water is going to diffuse out of them, and that can affect the structure of the cell. And that's going to be something that we investigate on the next couple of slides. So keep in mind that osmosis can be defined in two ways. It can be defined just in terms of water, so water will diffuse down its concentration gradient, or it can be defined by considering the more realistic scene that we find in nature, which is where water has solutes within it. And that is where water will diffuse from an area of low solute concentration to an area of high solute concentration. So let's watch this animation on osmosis then.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay, so let's investigate some of those terms that were mentioned in that animation then. First off, hopefully it helped understand osmosis in a bit more detail. It might be one of those things that's worth going over a couple of times though. Students do often seem to struggle with osmosis a little bit. I think it's the introduction of solutes that does seem to add confusion. So it's definitely worth going back over this a couple of times just to get it comfortable in your head, because it, like I said, it is a really important part of how animals are able to do what they do. So this process of osmoregulation, managing and balancing out our water and salt contents is incredibly important and necessary for life to continue. So definitely spend a bit of time having a look at it. Let's go through and investigate osmosis in a little bit more detail though, and go back over some of the terms that were mentioned in that animation. So he mentioned three terms: isotonic, hypotonic, and hypertonic. So let's talk about what they mean. So in this diagram, we have red blood cells or erythrocytes. So this is what, in the middle of the diagram here, this is what a healthy red blood cell looks like, or we have three red blood cells here. They form this biconcave structure. So they are almost quite plump. They have a high surface area for the diffusion of things in and out of them. And they're really important for carrying oxygen and CO2 around the body. And if you notice, they prefer to be in what is referred to as an isotonic environment. And if we look at the bottom here, what isotonic means is that the solute concentration will be the same inside the cell as it is outside the cell. So if you think red blood cells are typically traveling around in our blood plasma, which is largely water, then the solute concentration of that plasma, so the amount of stuff that's dissolved in it, the amount of sugars and salts that are dissolved in it, needs to be the same in the plasma as it is inside the cell, so in the cytoplasm of the cell. So what that means is you get no net diffusion of water in or out of the cell, but you will sort of get the movement of some water molecules, but you'll get no diffusion. So when the solute concentrations are equal, we refer to that as isotonic. What, however, would happen if we were to take those red blood cells and we were to pop them into a beaker of something like seawater? So seawater has a very, very high solute concentration. There's a lot of salts dissolved in that seawater. We would then call that seawater hypertonic. And we would say that it is hypertonic compared to the inside of these red blood cells. What that means is it has a higher solute concentration. So the seawater has a higher solute concentration than the inside of the red blood cells. So it is a hypertonic environment. And as you can see here, what will happen is because water will diffuse from an area of low solute concentration to an area of high solute concentration, it will move from the inside of the cell to the outside of the cell because it's saltier outside of the cell. So water will diffuse out of the cell into the surrounding water. And what that will do is it will cause the cell to crenate, or it will cause it to shrivel up and die. Although most of us here, or all of us actually, will be too young to have ever experienced this, until the dawn of refrigeration, so the fact that we all have fridges and freezers in our home now, which are really, really useful appliances for preserving food. Well, before those were available to us, one of the most common ways of preserving food was to salt it. So particularly with things like meat, you would pack it with salt. And what that would do is it would create this hypertonic environment, or one of the things it would do, it would draw water out as well, but it would create this hypertonic environment. So any bacteria or any fungi or other pathogen that landed on that meat and could otherwise start to break it down and start to consume it and spoil it, well, they would have the water drawn out of them because they were in a hypertonic environment. So the water would get drawn out of those pathogens and they would shrivel up and die. So that's why salting was, and still is in some parts of the world and some cultures, still a very, very important way for preserving food. So that's hypertonic then, a higher solute concentration. On the right-hand side here, we could imagine taking our red blood cells and instead of putting them in seawater, we put them into something like distilled water. So distilled water is essentially pure water. There are no, or certainly very, very low concentration of solutes, practically no solutes to speak of. So hypo refers to lower or less than. So a hypotonic environment is a low solute concentration. So in distilled water, we have a lower solute concentration than the inside of the red blood cells. So what's water going to do? It will start diffusing into the cell because it's going to go from an area of low solute concentration, so the distilled water, to the inside of the cell to an area of high solute concentration. And what that will do is it will cause the red blood cell to start swelling and eventually it will lyse or burst. So this again will cause the cell to die. You can hopefully see by this why I said that animals go to great lengths to osmoregulate, to regulate the water and salt balance within their tissues. Because if our cells are exposed to an area of high solute concentration, they will shrivel up and die. If they are exposed to a hypotonic environment, so a low solute concentration, they will swell, burst and die. So our bodies work incredibly hard to constantly maintain this isotonic environment where our tissues and the inside of our cells are kept at the same or very similar solute concentration. So hopefully that makes sense. What if you were a plant? Well, plants have a structure around their cells that we don't have as animals. And it makes the world of difference to how they osmoregulate or how they deal with water and solutes. And the structure that I'm referring to here is the cell wall. So we can see that in this diagram. This is the diagram that we looked at in the last session. So you have this cell wall that's made of cellulose that provides strength and rigidity. It also has an important role to play in relation to osmosis. So let's have a look at that on the next slide. If we look at the top part of this diagram, we can see the same as what I just showed you with the red blood cells. So these are 3 animal cells. In the middle here, we have an animal cell in an isotonic environment, so an isotonic solution, and we can see that the cell is normal, it's happy. On the left, so this is the other way around than on the previous diagram, but on the left here, we've got a hypotonic solution. So you can imagine that this is our distilled water. And just like the red blood cell, water is diffused into the cell. So we can see that water has gone from an area of low solute concentration to an area of high solute concentration. And that has caused the cell to swell and to burst. And on the right, we can see that the cell is in a hypertonic solution. And again, water will diffuse from an area of low to high solute concentration, so water diffuses out of the cell, causing it to shrivel up and die. Along the bottom here, we can see a plant cell in the same 3 environments. So let's start on the right hand side. So let's start with what would happen to a plant cell in a hypertonic environment. Seawater, for instance, if you were to take a typical terrestrial plant. Well, what you can see is the same thing will happen. Water will diffuse out of the cell, and that causes the plasma membrane to shrink and to shrivel away from the cell wall. And we call that plasmolyzed or plasmolysis. The cell will shrivel away and it becomes really, really limp, and basically the cell will ultimately die. If we put a plant cell into an isotonic solution, we can see that there's no net diffusion of water either in or out of the cell, but it can just sort of move freely in and out, but there's no huge amount of water movement. And that results in the plant cell becoming flaccid. So it's just kind of a bit limp. So this is not a good place to be for a plant cell. On the left-hand side here, we've got a plant cell in a hypotonic solution. So that is fresh water, so pure water, where there is a lower solute concentration, so a hypotonic environment. And what will happen now is that the water will diffuse into the cell. So it will go from an area of low solute concentration to an area of high solute concentration. And as the water diffuses into the cell, it causes the plasma membrane to push outwards. It starts to swell. So with the animal cell up here, that meant that the cell would swell and burst, but a plant cell has a cell wall. So now the cell is swelling, but instead of bursting, it's just pushing out against the cell wall, that rigid cellulose structure, and that makes the cell turgid. And this is a normal position, or the normal conditions for a plant cell to be in. It makes the cell turgid. This is something that we refer to as turgor pressure, the diffusion of water into the cell, creating this push outwards, this pressure that pushes outwards, meaning that the plant cell will be turgid. And that enables plants to sort of stay upright and to have these quite impressive structures without any sort of form of skeleton. So turgor pressure is really important for plant cells. And you would have probably, if you've ever kept a house plant, you would have noticed this. If you don't water it regularly, what will happen? Well, first off, it will start to go flaccid when it doesn't have enough water to keep diffusing in, and then ultimately it will dry out and die. You keep it watered, so if you've got a plant that you haven't watered for a little while and it's gone all flaccid, you can give it some water and quite quickly it will spring back to life. It will start to become turgid again as that water starts diffusing into the cells and pushing out against the cell walls. Osmosis then, it's very, very different in plant and animal cells, but it's still adhering to the same rules, that water will diffuse from an area of low solute concentration to an area of high solute concentration. So keep that in mind, because it becomes important in later sessions. So what we've looked at so far then are our examples of passive transport. So remember, diffusion, facilitated diffusion, and osmosis all occur completely free of charge. The cell does not need to expend any energy. So what happens if a cell needs to move something from an area of low concentration to an area of high concentration? So you're trying to move something against the concentration gradient. So if you remember earlier, I gave the example of the ball and the hill. So diffusion, facilitated diffusion, and osmosis are passive transport. They're the ball rolling down the hill for free. But now we're stood at the bottom of the hill and we need to get the ball to the top of the hill. We need to expend energy to do that. And this is the same place that we find ourselves with active transport. There are times when a cell needs to move things against the concentration gradient, and it will need to spend energy to do that. So active transport is the movement of substances against the concentration gradient from low to high concentration. Again, this will need special carrier proteins to do this, so this will not happen freely across the plasma membrane. And we typically have these highly specialized transmembrane proteins that have a very specialized 3D, almost kissing gate style action to them. And because we're going against the concentration gradient, the cell needs to spend energy in the form of ATP. And this ATP is generated within the mitochondria during the process of cellular respiration that we'll look at in a couple of sessions' time. So here I've got an example on this animation of active transport. Now, there are some other examples that I'll give you as we work through them through these sessions, but this is a particularly important one to the healthy functioning of our nervous systems. It's called the sodium-potassium pump. So have a look at it, pay attention to where sodium and potassium ions are moving and the fact that they are moving against their concentration gradient. And we'll actually revisit this in a later session when we actually look at the functioning of nervous systems. So this is actually a useful introduction to the process for that topic.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. In other words, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so hopefully that helped a little bit and made a bit of sense. Some of these are harder to understand and get used to than others. So typically I would expect osmosis and active transport to be the stumbling blocks. It is worth spending a bit of time in becoming used to them and getting a decent understanding, because like I said, they do underpin a lot of the topics that we're going to cover in later sessions. You know, it's useful if I'm mentioning osmosis or I'm mentioning active transport that you remember the keys of what I'm talking about there. So it definitely is worth spending a little bit of time on this. So what we can see on this slide then is just a summary of the modes of transport. So we have passive transport that can be just normal diffusion, so simple diffusion, including osmosis at this point, where things diffuse through the phospholipid bilayer. But then we also have facilitated diffusion, where things can still diffuse from an area of high to low concentration, but they cannot pass through the phospholipid bilayer, so they need specialized proteins, either channel proteins or carrier proteins, to enable them to do that. And then here we've got active transport. So I just gave you the example of the sodium-potassium pump that we find within our nervous systems. And the key to active transport is we are moving things against the concentration gradient. So from an area of low to an area of high concentration. And because of that, the cell has to spend energy. And that energy is called ATP, adenosine triphosphate. But don't go worrying about what ATP is and how it does what it does at the moment. This is something that I will cover with you in the not too distant future. But the key to it is that active transport means that needs the cell to spend energy. What we also find is that sometimes cells need to move larger molecules in and out of the cell. So we're not able to just move small molecules or small ions in and out. So when this happens, when the cell's trying to move something like certain proteins or carbohydrates in and out, they have to do something called bulk transport, the moving of these larger substances. And we can separate this down into 2 main terms. There's a process called endocytosis. So endo refers to inside, cytosis refers to the cell. So that is bringing something inside. And these are typically broken down into 2 main types. So phagocytosis that you can see here. So this refers to cell eating. So the cell will be bringing in some kind of solid. And you also get pinocytosis, which is cell drinking. So that's where the cell brings in some form of liquid. And then you get exocytosis. So exo referring to outside or out, cytosis cell. So this is where the cell removes things from the cell. So they move it outside. So let's have a little look at this animation, and hopefully that will help.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
So as per usual, let's do a finish off by doing a recap to see what you can remember from the session. So we've got 5 questions here. I'll read them out and then we'll pause the video for a bit and then give you a chance to answer them, and then we'll go through the answers together. So question number 1, what is the role of transmembrane proteins? Number 2, what is the role of cholesterol in cell membranes? Number 3, what is diffusion? Number 4, what is the difference between passive and active transport? And then number 5, what will happen to an animal cell that's placed in seawater? So have a go at answering those. There's some slightly trickier ones there, but give it your best shot, and then we will go through the answers together in a bit. Okay, welcome back. Hopefully you've had a chance to answer all of those questions. So let's go through the answers then. So number 1, what is the role of transmembrane proteins? Well, the answer is to enable the movement of substances in and out of the cell. So we could have written quite a bit more there, but transmembrane proteins form those channels and carrier proteins that will enable certain molecules and substances that can't get through the phospholipid bilayer. Well, it's those proteins that enable them to do that. Number 2, what is the role of cholesterol in cell membranes? It acts as a temperature buffer or fluidity regulator. So as long as you had something relating to temperature and fluidity, that would be ideal. So cholesterol sits between those phospholipids and prevents it from solidifying or liquefying, helping to maintain the fluid nature to the right level. Number 3, what is diffusion? Well, it is the movement of substances from an area of high concentration to an area of low concentration down a concentration gradient. Now, I would imagine that by this point in the session, you are fed up with me saying the word concentration. I have said it a lot. but it is incredibly important to sort of understand how it relates to all of this stuff and why it's important. Plus, I've nearly finished. So diffusion is the movement down that concentration gradient. Number 4, what is the difference between passive and active transport? Well, there are a couple of differences there that you could have mentioned. So active transport is against the concentration gradient. So we are moving things from an area of low concentration to an area of high concentration. So against the concentration gradient. And because of that, it requires ATP. The cell has to spend energy to do it. Passive transport is down the concentration gradient and occurs for free. And then number 5, what will happen to an animal cell placed in seawater? Well, water will diffuse out of it. So water will diffuse via osmosis. So it will move from an area of low solute concentration to an area of high solute concentration. And that will cause the animal cell to shrivel up and die. Okay, so hopefully you did okay with those. As always, don't worry if you didn't do too well or you got a couple wrong. This is just an opportunity for you to highlight the areas or the parts that you are comfortable with, but also the parts that you need to go back over a few times to become familiar with them and to get to increase your understanding of them. Okay, so that brings us to the end of this session then. So let's just do a quick summary. So hopefully by now you should be able to identify the key components of the cell membranes. You should be able to discuss the roles of each of those membrane components. So remember, we're talking about here things like the phospholipids, the cholesterol. So we can see those here. You've got the transmembrane proteins, the peripheral proteins. You have the glycoproteins and the glycolipids. And then also you should be able to compare and contrast, so identify the similarities and differences between the modes of transport across cell membranes. So that is simple diffusion, facilitated diffusion, osmosis, and then active transport. Okay, so that brings us to the end of this session. Hopefully that was useful, and I will see you in the next one. Thank you very much.